Well, thank you very much, Neil, for that introduction. And I'd like to begin just by thanking the Microbiology Society for this award. It's a tremendous privilege to stand here and speak to you all this morning. And as Stephen Baker said last night, actually the really exciting thing is joining the line of people who've had the award. Now, actually, I took a look, and the first lecture to commemorate Marjory Stephenson was actually held in 1953. So since then, a whole line of folk have each time stood up and presented their current work, but with one eye to the past, to Marjory Stephenson. And actually, I think that's a really important thing to do, because, in my opinion, you can only really fully appreciate the microbiology present if you have an appreciation of microbiology past and our microbiology heritage. And actually, you could argue that this is a key role for learned societies such as the Microbiology Society, or rather, I should say, it's one of the many very, very important roles for learned societies. Okay, so Marjory Stephenson was a member of staff at the Department of Biochemistry at the University of Cambridge for 30 years, almost 30 years, up until her death. And it's generally agreed that her principal contribution was to identify and investigate many enzymes and pathways in microbes. And actually, a lot of her discoveries underpin many of the things that we take for granted, actually, these days. But as Neil said, she discovered something else. And what she observed was that the measured, for many of the enzymes she studied, the measured activity varied enormously according to the conditions in which the particular microbe was grown. And she called this enzymatic adaptation. Now, it's quite clear that she fully appreciated the biological significance of this, because, of course, it allows microbes to adapt and to respond to their environment. But, of course, she had absolutely no idea what the molecular mechanism was. And this problem was taken up by a young Frenchman called Jacques Monod, shown here in the middle. And Monod worked at the Pasteur Institute in Paris. And it's worth remembering that when Monod started, his starting hypothesis was actually that enzymatic adaptation was due to something taking place at the level of the enzyme. Now, I imagine that most people in this hall know that subsequently Monod showed that this wasn't the case, and actually enzymatic adaptation is due to issues of gene expression. And together with his boss, André Leboeuf, shown here on the right, and his colleague, François Jacob, they established the operon model of gene expression, in which a gene has to be transcribed, and this transcription is done by an enzyme called RNA polymerase that starts at a specific location. It's called the promoter. And because it starts at a specific location, that means it can be regulated at that location. And sure, you all know that probably their most renowned discovery is that this regulation can be done by a repressor. So basically, the repressor just stops the RNA polymerase from doing its stuff. And some years later, it was discovered that at some promoters, there wasn't a repressor, but the regulation was due to an activator. And basically what the activator does is it turns on the promoter so that polymerase can transcribe the unit. And because we now know that there are many bacteria where there are hundreds of transcription factors, activators and repressors, and these are responsible for orchestrating, if you wish, the responses of the the bacterium to environmental changes. And of course, many of you will know that in the paradigm system that Monod and Jacob studied, the Escherichia coli K12 lac operon, there is actually both a repressor and an activator. So you get two transcription factors functioning there. The repressor, of course, is the lac repressor that responds to the presence or absence of lactose in the medium. The activator is a protein called cyclic AMP receptor protein, CRP. Neil just mentioned it. And the activity of CRP is triggered by cyclic AMP, which is a second messenger that reports the presence or absence of alternative nutrients such as glucose. Now, I've always been totally fascinated by transcription factors, both activators and repressors, how they work. They always seem to me to be wonderful devices and worthy of understanding. But I have to confess that when started working on transcription factors, we didn't think too much about impact. I mean, impact wasn't very trendy then, and we just studied them because they were beautiful and we wanted to understand them. However, with the passage of time, it's become clear that there are really two areas where studying transcription factors has big impact. So one is in biotechnology and synthetic biology, where transcription factors really are the nuts and bolts. And the other is in the study of pathogenic bacteria, where it's quite clear that the expression of many virulence factors is controlled by transcription factors, and often these are bespoke transcription factors for the virulence factor. And of course, this raises a really interesting possibility that you could devise antimicrobial strategies to hit those bespoke transcription factors. And of course, the consequence of this would be that you disarm the bacterium rather than killing it. And in many circumstances, that would be a good thing. But today, I'm not going to talk about any of that. What I'm going to do is I'm going to focus actually just on activators and how activators activate transcription. And the talk's going to come in three halves, three parts. So I'm going to give you a fairly lengthy introduction to bacterial transcription and its initiation. I'm going to tell you what we learnt from CRP, and I'm then going to tell you how we learnt something different from CRP's cousin, FNR, and its helpers. So, the introduction to bacterial transcription. So all bacteria actually face a problem when it comes to transcription, and the problem is this. On the one hand, you have to ensure that you copy the DNA into RNA, but on the other hand, the bacterial chromosome has to be compacted to make it fit inside the DNA. And these are essentials because you have to do these two things. But the problem is that the two things actually often, in fact mostly, run counter to each other. So for compaction, obviously we know that the compaction is done by negative supercoiling, but it's also done by a family of proteins. These are small proteins that are called nucleoid-associated proteins. So these are proteins such as IHF, FIS, HU, HNS, a whole bunch of others. And these proteins are mostly quite small. They're quite promiscuous in how they bind, so they bind all over the chromosome. And when they bind, they're also quite abundant, because they need to be to cover the chromosome. And when they bind, they actually induce conformational changes in the DNA. Consequence of that is to compact the bacterial chromosome. So, of course, you can see quite easily that doing that is probably not very good for transcription. Now, because set against that, pushing in the other direction, you've got the DNA-dependent RNA polymerase. So let me tell you a little bit about that. This is just a sketch I downloaded, I think, from one of Arkady Mustayev's papers. So the polymerase is a little molecular machine. It's wonderfully clever. What it does is it makes RNA copying a DNA template strand. In order to do that, you have to open up the DNA. So it has to be a local region of melting, and basically what the enzyme does is it threads the strand that's going to be copied through the active site that's in the middle of the enzyme, and the active site then makes RNA, and the RNA is threaded out and it just moves along doing that. Now, all bacterial RNA polymerases are multi-subunit. They consist of several subunits, so there's just another little sketch that illustrates the subunit structure. So here it is. The two big subunits that form the cleft are called beta and beta primed, and often people refer to that as a crab claw. And the beta and beta prime subunits are held together by alpha. Actually, there are two alpha subunits, and it's a little bit complicated because each alpha subunit has two domains. So the N-terminal domain of each alpha subunit does the holding together, and the C-terminal domain, I'll come on to that in just a moment. Now, this form of the RNA polymerase is known as the core enzyme, and it has an important property, or an important non-property, which is that it cannot find promoters and initiate transcription. So in order to find promoters and initiate transcription, you need another subunit. That subunit's called the sigma factor, and the job of the sigma factor is to find the promoter and then to open up the DNA and make sure that the template strand goes into the active site. And all bacterial sigma factors are multi-domain proteins. They consist of two or more domains hooked up by flexible linkers. So the major sigma factor of E. coli is called sigma 70. It consists of 4 domains, and by far the most important domain is domain 2. And the reason for that is that what domain 2 does is it almost on its own, single-handedly, it melts a local bubble in the DNA at the start of where the transcript is going to be, and it arranges for the template strand to go in the active site of the enzyme. Of course, in order to do that, it has to be held in place, and that job is done by domains 3 and domain 4, and domain 3 and domain 4 recognize different promoter elements. You've probably heard about promoter minus 35 elements, so it's domain 4 that recognizes the minus 35 element. Now, in order to make this a little bit clearer, I've pinched some rather nice diagrams from Ramesh's lab at Imperial, and Ramesh kindly gave me these. And I should say that these diagrams that you're going to see are based on some absolutely mind-bending structural biology from 3 labs in the States, Seth Darst, Richard Ebright, and Tom Stites. So I'm not going to go into the molecular details, just take a look at the diagram. So here's an RNA polymerase that's got to a promoter. Here's the active site. The active site's empty, okay? And the DNA is still duplex. But domain 2 is there sitting ready to pounce. And it's held in place by domains 3 and domain 4 that are bound to their cognate elements, to cognate elements at the promoter. Now I press the button, so now domain 2 has kicked in. It's now melted the DNA. It actually holds the non-template strand. The template strand goes into the active site, and what now happens is we're set up to start the transcript. Okay. So the bottom line of this, very important conclusion, is that RNA polymerase containing a sigma factor actually is perfectly capable of doing the business, of finding a promoter and initiating transcription. So you ask, well, what about promoters that are dependent on activators? Well, clearly at those promoters, there must be something wrong. So either the polymerase can't recognize the promoter, or when it does, it mispositions the polymerase. When the polymerase binds, it's mispositioned, or maybe sometimes there's something wrong with this, so that domain 2 can't do its stuff. And when we started our work, there were really 2 models for how activators worked. One was that the activator bound at the target promoter and passed some kind of magical conformational change through the DNA that set up the promoter so that the polymerase could do its stuff, or that the activator made some kind of direct contact with the RNA polymerase. And I decided that I wanted to work out which mechanism was operating. We were going to solve this problem, and we were going to solve this problem using the cyclic AMP receptor protein from E. coli K12. And this was a good choice because at the time, the high-resolution 3-dimensional structure of CRP bound at a target had just come out from the Tom Stites lab. And here it is. Again, it's the most beautiful structure. CRP is actually functional as a homodimer, two identical subunits. But in the structure here, the two subunits are coloured differently for clarity. And what you see, let me just talk you through the structure. Each subunit has a cyclic AMP nestled in the middle. And the cyclic AMP sets up the conformation of the CRP such that the recognition helix in each subunit penetrates a major groove in the DNA. So the two subunits of the dimer penetrate two adjacent major grooves. And of course, once there, you get the sequence-specific contacts that dictate the CRP goes to specific sites. And that really is very, very beautiful, but actually that wasn't the headline news in Stites' paper. The headline news was that when the CRP bound, it actually bent the DNA quite violently. Now, this is the title of the Stites paper. It actually turns out when you do the measurements in solution, the bending's more like 140 degrees, 150 degrees. So it really looked at this moment that CRP was activating transcription by changing the conformation of the DNA target at the promoter. But it turned out that that wasn't the case. And what changed our minds? So that the crucial thing that changed our minds was the identification of some CRP mutants that were unable to activate transcription. The identification of some CRP mutants that were unable to activate transcription. Now, of course, it's easy to get mutants that are defective, but these mutants were special. And they were special because, first of all, the mutant CRP was produced normally. Secondly, it folded normally. It bound cyclic AMP normally, bound to DNA normally, even bent the DNA normally, but was unable to activate transcription. And I think actually Karen Chapman first got the first of these mutants, but we got some, Richard Ebright's lab got some, and Bill Resnikoff's got some. And what we all agreed on was that these mutants all carried substitutions, these mutant proteins all carried substitutions in one location. This location was a little beta turn just off the DNA. And we called this And the interesting thing was that the substitutions were all in amino acids where the side chains pointed outwards. So we called this an activating region. And Ebright and myself were able to show, at least for the LAC promoter, that you, in order for CRP to be active at the LAC promoter, you only needed the activating region in one of the two subunits, and actually it's a downstream subunit. So on the basis of all this, it really looked as if the crucial thing that you needed to activate was not to bend the DNA, but was the activating region. And it really looked as if the activating region must be making some sort of contact with something else. And of course, that something else was, of course, the RNA polymerase. So this was the cue for a massive number of experiments and person years involving my lab, Annie Kolb's lab in Paris, Richard Ebright's lab, Akira Ishihama in Japan, Rick Gorse. And I'm not going to go through any of the data. I'm just going to show you the model. And the model is, yes, indeed, the activating region is a contact patch. We call it activating region 1 for reasons that you'll understand in just a moment. And the bottom line is that the cognate contact site for activating region 1 in RNA polymerase is in the C-terminal domain of the RNA polymerase alpha subunit. And you need to make this contact for transcription to be activated, at least at the LAC promoter. And actually, this was a really nice result because at the time, several people, including ourselves, had noted that there were a whole bunch of promoters where the DNA site for CRP was just slightly further upstream. And we were easily able to show that you needed the same contact site at these other promoters. And of course, that was instantly explicable because, of course, alpha CTD is linked to alpha NTD by a flexible linker. So the linker, of course, is there to allow some sort of flexibility. Anyway, that was all jolly fine. But of course, the question, there's a contact. the question was then, what did the contact do? And this is where Richard got the answer. So by using structural biology. So what he did was Richard determined the structure of CRP and alpha-CTD on their own, but also in combination at a promoter. And Ebright's conclusion is very, very clear. That is, the conformation's the same. Okay, so there's actually no, when the contact's made, there's no change in conformation of the other partner, and this is being found, I mean, many ways. And so the conclusion was that the contact is just Velcro. And so basically the bottom line is that the promoters that are dependent on CRP are defective in some way because the polymerase can't bind, because some of the promoter elements are not sufficient, and basically what you do is the CRP just offers a little extra contact that then glues the polymerase to the promoter and presumably positions domain 2 such that it can do its stuff. Now, it was at this point that we ran into a bit of a problem. And the problem was that by this time we'd got something like 100 different CRP-dependent promoters. And many of them, the DNA site for CRP was actually too close to the promoter elements for this sort of arrangement to take place. And so once we realized this, we set to work, and it was pretty much the same consortium, but this time with Jim Hoggett from York I did. And what we showed was that, yeah, even at these promoters, CRP does the same sort of thing, but this time, because there's no room for the alpha actually gets kicked, the alpha-CTD gets kicked out and binds upstream. And now the downstream subunit of the CRP dimer is pushed up against other parts of the RNA polymerase. And actually, to cut a long story short, it turns out that there are 2 more activating regions, and they're displayed in this subunit here. So one is activating region 2. This was discovered by Richard, which makes a contact with alpha NTD, and the other is activating region 3. These are separate patches on the CRP, and activating region 3 contacts sigma domain 4. Now, it turns out for most of these promoters, activating region 2 is the major determinant, and region 3 only plays a very, very small role. But nevertheless, there are two contacts. And actually, to make these 2 contacts, you have to really position the CRP very, very precisely at these promoters. And again, these things work just by Velcro. Okay, so the conclusion was that basically CRP could activate transcription by 2 different mechanisms, and these became known as class 1 and class 2. It was at this point, actually, that the penny dropped. We had the moment of clarity. And I realized that actually lots of other transcription factors played exactly the same game. It wasn't just CRP. So actually, I need to tell you that CRP is a member of a big family of transcription factors spread across a whole bunch of different bacteria, and they all played the same game. The Araceae family members, they play that game. Response regulators, a whole bunch of virally encoded transcription activators do exactly the same, and probably ones that haven't been examined yet play the same game. But actually, what we found was for many of these other transcription factors, they could either do class 1 or do class 2, but couldn't do both. That CRP was sort of unusual because it had different activating regions, so it could do both, whereas most transcription factors could only do one. And that was actually quite a good, important realization, because it explained something that had been puzzling us, and that is that many people had noticed that many activator-dependent promoters in E. coli, and actually now in lots of other bacteria, you don't just need one activator, but you need 2. Okay, so many, and the biological relevance of this is clear. It's just to couple activity to different signals, and it had been noticed that at many of these promoters depended upon 2 activators. One is in a class 2 sort of position, the other's in a class 1 position. So it was easy to see that these promoters must just need an extra dose of Velcro if you wish to work. Now, unfortunately, I just don't have time to go. Actually, it's a little bit more complicated than I'm letting on. So I don't have time and you don't have the inclination to listen to the boring details of these promoters, but it's all in our annual review. I'll just say one thing about this sort of promoter, and that is, the big question is, if you're going to have a promoter that's dependent on two activators, you've got to make sure that one of them can't do the business, right? Okay, so how do you do that? And it turns out, at least for CRP, that the trick is to misplace the CRP by one base. Okay, so what you do is you misplace the CRP. That then confers codependence on a second activator. It is fearfully cunning, as Blackadder might have said. Okay. Right, so let me now just give you some conclusions from this part of the talk. Most activators, I suspect it is most, work by using one or more activating regions to recruit RNA polymerase to target promoters, and the interaction serves as Velcro. Now, I have a little confession. When we realized that it was just Velcro, I was a tad disappointed, because in my heart, I had always nurtured the idea that these activating regions were like fingers that would press the magic spot on the polymerase, and pressing this button would open up the polymerase and switch it into some sort of excited state, so it would be raring to go. So it was a bit disappointing to realize that it was just Velcro. And actually, it's just Velcro at most activator-dependent promoters. And the reason's very, very simple. You know, when you think about it, it was so stupid to think it could be otherwise. The reason's so simple. It's all because what I told you about domain 2. All you have to do is position domain 2 of sigma, and domain 2 will do the rest. Okay? So you don't need the magic button, because domain 2 will do it for you. And of course, this applies to all sigma factors except one family. There's one family of sigma factors, the sigma 54 family, that don't have a domain 2. Okay, now this is not my work, but it's really germane to note this now. So it's been known for years that RNA polymerase carrying sigma factor that is related to sigma 54 requires a special activator. These special activators are called enhancer binding proteins, and these special activators do press a button. And absolutely amazing work from Xiaodong Zhang and Martin Buck at Imperial have shown that sigma 54, the activators that interact with RNA polymerase sigma 54 do press a button, and they actually literally caress the RNA polymerase and induce it to do its stuff. And this actually costs ATP. Okay, conclusions number 2. There are 2 major modes of activation by recruitment. Many activators have just class 1 activating regions, others have just class 2, but some, like CRP, have 3. So, you know, a germane question you might ask is, well, what about other members of the CRP family? Do they have 3 activating regions? And let me answer that by telling you about FNR. So in E. coli K12, there are 2 functional CRP family members. One CRP itself, the other is FNR. FNR is a transcription activator that activates in response to lack of oxygen, so it's a global regulator. And work done in my lab together with John Guest and Jeff Green showed that actually FNR has a very, very strong activating region 3. It has really hardly any activating region 2, and it has a sort of decent-ish activating region 1. So the conclusion is, yeah, FNR does have the 3 activating regions, but the emphasis or the weight varies. And actually, this applies to most members, as far as we can see, of the CRP family. So you know, it's as if there was this structure, the CRP structure, and it's almost as if the activating regions were painted on at a later stage. And I'll return to that in a moment. Okay, so the summary from this part of the talk is that studying CRP taught us how activators activate transcription. But it turns out that that's only half the story. because there's another half of the story that I'm going to tell you now that doesn't involve activating regions, but involves nucleoid-associated proteins. And we got into this by through my lab neighbour, Jeff Cole. So Jeff has a lifelong interest in nitrate and nitrite metabolism. And one of the genes that he's particularly, operons that he's particularly interested in, encodes for an NADH-dependent nitrite reductor. So that's exactly the sort of enzyme Marjory Stephenson would have been interested in. I got interested in it because Jeff told me that this operon is expressed, is regulated, it's very tightly regulated from a single promoter, and it's dependent on it's very tightly dependent on two physiological signals. One is the lack of oxygen, which is interpreted by FNR, and we were able to show that FNR binds to a single DNA site at the near promoter. It's a class 2 sort of position, and the other signal is nitrate, actually nitrite as well, and that's interpreted by NARL. And we're able to show that NARL binds to a single DNA site, actually at a class 1 position. So when I saw this, I thought, oh, chic, it's one of those promoters where you have the two activators, functioning independently, like I just showed you. But it turns out that's not the case. A very simple experiment shows this. So what we did was we just replaced the upstream sequence with some unrelated sequence, so NARL couldn't bind. And when you do that, because the prediction is the promoter should just drop dead, but it didn't. It was fully alive, FNR dependent, but fully alive. And this was the trigger for doing a whole bunch of experiments that, again, I don't have time to go into, but it involved a whole bunch of PhD students and some postdocs, and we ended up with a model, and here's the model. So this is the situation in anaerobic conditions with no nitrate. So FNR binds to its target, and it's ready to do its stuff. It's ready to activate, but it can't. And it can't because of NAPs, nucleoid-associated proteins, that bind upstream. And there's FIS that binds to a single site, and two molecules of IHF that bind upstream too. And when NARL comes along, what happens is NARL actually kicks IHF off one of the sites, and this busts the repression. Now, I put up a picture of Doug Browning here because this is really his story, actually, and he's done a lot of the stuff. And I'll show you just a couple of his results. So let me just show you this NARL kicking IHF off. This is a footprint. So, in a footprint, a DNA is one footprint. What you do is you attack a piece of DNA with DNAs, and you get a series of bands, and you see blank bits with no bands where the protein protects. Let me just talk you through this. It's a wee bit complicated. So here's protection by NARL. Here, very nice protection, single site here. I think it's lane 5, yeah. Here's lane 5. Here's protection by IHF on its own. So it's quite a long footprint because there are two sites, called site 2 and site 1. Now, if you pre-bind IHF and then add NARL, what you see is the NARL binds immediately. The IHF is kicked off. You can see it's kicked off site 1, but it remains on site 2. The reason why it's kicked off so efficiently is that NARL's a major groove binder and IHF's a minor groove binder. So the conclusion from this is, NARL displaces IHF. And this is actually activation by anti-repression. Okay? So what we want to argue, I'm going to develop this now in this final 5 minutes of the talk. What we want to argue is this is a more ancient way to activate than activating regions. And why do we think this? Well, we know that DNA compaction and transcription are essential, whereas regulation isn't essential. So there must have been compaction and transcription before regulation. And so it's easy to imagine that primitive activators might have worked just by pushing NAPs out of the way, like this. Okay, so that's the hypothesis. So the question then, of course, becomes, well, where did the activators we see today come from? And actually, you don't have to look very far to get a suggestion. And here's the suggestion. So we're now back with the near promoter. And remember, I told you that NARL functions by kicking off IHF from one site, leaving IHF bound at the other site. So what Doug decided he was going to do, he was going to look at the the effect of IHF bound at site 2. So the way he did this was by making some mutations in the DNA site for IHF, and it's a little bit complicated. When I press the button, you can see it's a bit complicated. So there's going to be a mutation that makes the site consensus, and that's marked in green, and the mutation that destroys the site, and that's marked in red. Now, I, of course, told Doug that this was a total waste of time because, of course, IHF2, like all NAPs, represses. But actually, I was wrong, and here's the result. So what you see, the green mutation, with the green mutation that improves binding to site 2, the activity actually goes up. And if you destroy the site, the activity goes down. So the conclusion from this is that IHF binding at IHF2 actually activates. Okay, so this is telling us something, I believe, really important. This is telling us that actually NAPs can be positioned such that they activate. And actually, Rick Gorse and Reed Johnson have shown that that some NAPs, or FIS in particular, can actually acquire an activating region. So if you buy these two, well, one's a fact, the others are. Yeah, both of them are facts. If you buy these two facts, it's only a hop, skip, and a jump to the hypothesis that maybe transcription factors evolved from NAPs. So the question then becomes, well, can we find some evidence for this hypothesis? And this is where this wonderful technique called chromatin immunoprecipitation comes in. Okay, so I'm going to show you a chromatin immunoprecipitation experiment. So the point about chromatin immunoprecipitation is that it allows you to interrogate a DNA-binding protein. It allows you to identify all its targets across the genome, independent of function. Okay, it just says, does it bind or does it not bind? Right? Okay, so Dave Granger, who was in my lab some time ago, he actually pioneered the use of CHIP for E. coli. One of the first profiles he looked at was FIS. And just to remind you, so the x-axis is the whole E. coli chromosome with the regular coordinates, and the y-axis is the intensity. And what you see is what you'd expect. FIS binds pretty much everywhere. There are some peaks because it has preferred sites, but it binds pretty much everywhere. And the crucial point is that it binds not only in intergenic regulatory regions, but it also binds within coding regions, because that's what you'd expect of a NAP, whose primary job is to fold up the DNA. Okay, so one of the other proteins Dave did was CRP, and here's the profile for CRP, that well-known model transcription factor. And as you'll see, it looks pretty similar. Okay, it actually looks pretty similar. I should say that this experiment's just recently been repeated in Ashwin's lab in Bangalore, and he using the new sequencing technology, and he gets pretty much the same result. So the conclusion from this is that CRP binds in coding and non-coding sequences. That's what this tells you. And at many targets where CRP binds, it doesn't activate transcription. So the conclusion is actually our friend CRP is actually a NAP. A nucleoid-associated protein. Now, if you buy that, you don't have to buy it, but if you buy that, it explains why CRP bends DNA and binds promiscuously. And, of course, it suggests that the activating regions are really just bolt-ons. And this accounts for each CRP member having different activating regions, because they were just bolted on at a later stage, presumably to equip the regulator to do its stuff. Okay, so now let me just wrap up give you a little summary of this second part of the talk. So the conclusion is that the summary is that by studying the E. coli near operon, we discovered that transcription activation isn't just a question of activating regions, and you can activate by pushing NAPs, by remodeling the local DNA environment and pushing NAPs out of the way. And I should say that we've shown, we have several instances now where NARL does that, and of course other investigators, in particular those who work with HNS, have found pretty much the same thing. So now let me just say a word about ancient and modern. So the contention is that that pushing NAPs out of the way is the ancient method of transcription activation, doesn't involve activating regions. And the speculation is that modern transcription factors with their precision activating regions that target specific patches on RNA polymerase, these were late arrivals, if you wish, at the molecular biology ball. And of course, the second speculation is that everything came from NAPs. So presumably, once NAPs had acquired activating regions, they then, some of them unbecame NAPs. They lost the ability to bind promiscuously, but some proteins like CRP in E. coli K12 retain that. And a couple of years ago, Sandhya at the Indian Institute of Science in Bangalore and myself wrote this little opinion piece, Evolution of Bacterial Transcription Factors, How Proteins Take on New Tasks that Do Not Always Stop Doing the Old. And actually, when we written a bit of a light-hearted piece, I was rather expecting some young sequence jockey to call me up and say, look, Steve, this is just not on. My sequence data shows that you're wrong. It hasn't happened yet, but the target's still there. And who knows? Maybe who knows? Maybe somebody with one of these guys might actually find some supportive evidence for this. Okay. Of course, so my contention, of course, just going right back from the beginning, is that, of course, you need to understand the past in order to fully appreciate the present. And by the way, while we're on the question of the present, of course, if you're a molecular microbiologist, it's fantastic to be in the present because, of course, there are always exciting new things to do. So at the moment, my lab's involved in several new angles on regulation, because of course you're never finished. One is how actual location within the folded chromosome affects regulation. The other is the extent to which bacteria use epigenetics to control their gene expression. So it's always, the present is always very, very exciting. Now, I can see that it's almost time for coffee, but before I let you go for your coffee, I just have to say a few thank yous. So the first thank you really goes to our funders. And over the years, the work's been funded by many, many external funders. But I would just like to mention the BBSRC, who've stuck with us right from the very, very beginning, even when they were called SERC. I'd also like to mention the University of Birmingham. You know, it's very easy to forget how much support one's employer actually provides for one's research. For many people, that's because they spend their time moaning, right? But actually, University of Birmingham has been unbelievably supportive and has actually put a lot of resource into microbiology. And Neil just told you about the Institute of Microbiology and Infection. And if you look in the booklet, you'll see we're organising a wonderful symposium in September. So I encourage you to come along to that. Now, the other people I need to thank are, of course, the people who did the work. And ideally, what I would do is I would want to put up the name of every PhD student and postdoc and technician who'd worked in my lab and thank them for their just amazing dedication, enthusiasm, inspiration. But alas, PowerPoint won't allow that because there are just too many names. So what I've done is I've just picked the staff, either students or postdocs, whose direct results fed into the story I've just told you. And I've tried to organise them in sort of some kind of chronological order, starting from Kevin Gaston and Andrew Bell right at the start, who first really got into activating regions, right through to Dave Chisman and Far Ruanto, who did some stuff on NAR-L as a class 1 activator. I'd also like to thank two technicians, Chris Webster and Rita Godfrey. Chris particularly, because she was my pair of hands for 25 years and amazingly lived to tell the tale and is now happily retired. And finally, we've had some fantastic collaborators. And again, I've tried to organise them in some sort of chronological order. And finally, I'd thank you and hope you enjoy your coffee. Thank you.